So moving briefly, I'll be talking about some microbiology laboratory tests. Microbiology laboratory test. So we'll be talking about colorimetry. This was a full exam question. They said students should write short notes on what colorimetry, talk about bioluminescence, talk about electrical impedance, talk about the gas liquid chromatography. Now, if they give you a lot of lab tests like this to do, well, you don't really have time. So I would have actually just advise you just state like the use and probably like just state the principle briefly to me. You don't have to go and start using all the testing all those procedures and all of that, okay, because there's no time in an exam situation. So first one is colorimetry. For colorimetry, we said that it relies on color changes of pH indicators in the media just to indicate the presence of metabolic end products. So you are targeting the metabolic end products. So a change in wavelength of the light transmitted through the cuvette or well is measured by photosensitivity by an photoelectric cell. Okay, so this is the dictator. This should be the photoelectric cell. So now it is the measurement of colored end products that indicates if a particular bacteria is present or not. Now that's just the principle you have stated. Now filters are used to absorb out certain wavelengths of light for dictation of color changes. Okay, so probably you might just put some examples of bacteria here. All right, it can be used to dictate presence of blah blah blah, Therefore, bioluminescence now. So bioluminescence is that fireflies' tails produce a glowing cold light, okay? And this same cold light is what you can see in some chemical reactions. So if fireflies are able to produce some one or two lights, some chemical reactions also produce light. So use bioluminescence to actually dictate all of this. So there's conversion of substrate luciferin to oxyluciferin and light. So it's catalyzed by an enzyme known as luciferase. So there's accompanying dephosphorylation of ATP to ADP. So you just write the equation down here so that you make the lecturer happy. So luciferin is converted to oxyluciferin and light to be catalyzed by luciferase, right? ATP will be converted to ADP. Now I didn't have it's supposed to be like this, so you write ATP here, all right, you write ADP here. You know how they do in that biochemistry stuff. So we said that light generated from this reaction can be measured directly using a luminometer. So amount of light measured in photons produced by the reaction in excess of luciferin is directly proportional to the amount of energy that was generated in the solution. Okay, so ATP is present in all living cells, including bacteria. So when microorganisms grow in culture media, luciferin can be added, and the energy produced by the bacterial cells convert luciferin to oxyluciferin and light, and the amount of light generated is now measured. Right, so it will now show a proportion of the energy consumption of the bacteria. You get, so it's helping dictating different bacteria. Electrical impedance is very simple. First of all, you can culture yeast or bacteria, then you pass an electric current through the medium. So the amount of electrical current trapped by the culture medium is referred to as impedance. It didn't allow it to pass, you impeded it. So clinical use is not yet established though. The last one is chromatography, gas chromatography. So for gas chromatography, we said that movement of particles is based on their size, their molecular weight and charge. Now a liquid sample is passed through a matrix, that differentially separates its constituents. Okay. Should be the matrix, separate the constituents. So the volatile components are carried along with the flow of especially what prepared heated gases through a narrow column packed with resin or some other materials that slow down the components based on their size, the molecular weight, and charges. Okay, so it's useful in dictation of anaerobic bacteria like Clostridium difficile. So that's it for this I think it's 4 laboratory tests. Okay, 4.